Good. I'm learning. The new assignment, yeah, Sean. When you do the first exam that morning, that the ones are due, then the other ones pop up. Then they're available for you to do. I don't want people jumping too far ahead. I mean, I like I said, those assignments are, you know, just intended to get you ready for exams. But is that what you're asking? Right, and so that's where you go to that spreadsheet that's on the assignments page. It's got like the chapters, tells you the assignments, and then it kind of blocks it by the exam days. You know, it says chapters 22 to 26, these assignments, there's these different kinds, and then they're all due on exam one day. So anybody got questions on when we're talking about this? If you got any other questions, go ahead and interject in those right now, no problem. But anybody got any questions about when we talk about variation? Of course, variation, like I said, is crucial to any of this whole thing of natural selection, because once again, the whole idea behind natural selection is if you take Darwin's natural selection mechanism is if you take a group of critters of the same species and they're all breeding, but some of them have some type of advantage, right? So that advantage means they have greater fitness, and we'll talk next chapter, right? A little bit more about fitness, I believe, in the next chapter. And fitness is basically a relative term that says, I live longer and have more offspring than you, therefore I'm more fit. So it's like a multiplication thing in math. It's how many offspring you have per year times how long on average you live, and all that sort of stuff adds up to very simple math, right? The longer you live and the more kids you have, ding, 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 you put that into your calculator, the more that your genes, your genetics, or parts of them, obviously, because we're not asexual, are passed on into the next generation, right? So if you're doing a lot of reproducing, because now one thing you might not know is the way metabolism kind of works in living organisms. It's kind of like a bank account. Actually, it's kind of like bills. Sorry, scratch that. It's kind of like bills, kind of like paying your bills. You bring in X amount of calories and energy, and X amount of calories have to go out. Like they have to help you walk and do the things you don't even think about, in addition to the things that you do think about. Reproduction is kind of one of the things that oftentimes are the last thing paid, okay? It's important to stay alive. Like, for example, some critters, if they've had a rough year of resources, they'll skip a year of reproducing. They won't reproduce this year. Critters that reproduce that, seasonally or something like that. Because reproduction is like a luxury to them, right? So organisms, in terms of reproduction, that's going to be an awful lot of effort put forth. So the whole idea is you're passing out your genetic information into your offspring. And it's something about you having an efficient way to process resources means you have a lot of money left over, a lot of energy left over to do this reproductive things. You can make lots of flowers, lots of pretty flowers, lots of big flowers. You can have a really pretty shiny coat if you're a horse, or run faster, or really bite like hell worse than the other stallions and fight better, and maybe among the lizards and there's some difference based on your coloration. Maybe females really like the red-bellied guys over the white, something like that, right? So what we're going to continue on talking about today is this whole idea of variation. But now that we've talked about, what we mean by it, the kinds, how we measure it, I want to now start talking to you about snowmen. No. Remember, of course, nature and nurture. There's the idea of genetics and its relationship with the environment. That's something that we kind of take for granted now, but when I first started teaching this class, that was something we were like, yeah, maybe, but now we're fairly well convinced that there's a lot of evidence out there, a lot more genes that we find that it depends on the environment they're in. I think I was telling you all about the whole idea of sea turtle nests. You know, the fact that eggs on sea turtles, the sex is really determined by the temperature where the eggs incubate. So those that further down in the sand are cooler, and I don't remember what sex it is, but they tend to be more of one sex versus the other. So, you know, the environment and genetics is really fluid, especially in some critters like these caterpillars here. But here we are. We're going to talk about where we get genetic information, or I'm sorry, variation now, how we form genetic variation. And we're going to go through 4 ways in which that happens. First of all, making new alleles, forming new alleles. So genetic mutations, right? Like changing the A's and the G's, changing that sequence of letters in the DNA. But like I told you before, you know, you got to really be careful with that. Remember I said it was like poking the devil with a short stick, right? My mother used to say when I was getting close to getting in trouble, if you start monkeying with the DNA, right? Now with those of us who have introns, right, the parts that aren't read, not big a deal. When you start messing with the exons, and evolution is only going to be working on the stuff that's in the exons, right? The introns aren't expressed. Nothing for Mother Nature to select, right? So this formation of new alleles, if that occurs in the exons very often, I always want to reinforce that, and I'll say it a million times this semester, ends up dead, muerto, dead, right? If you mess with the DNA, you screw it up. That's because many times changes to phenotype, changes to the way an organism expresses that gene, changes to that aren't good. Why? Because they're good right now. Don't fix things that aren't broken. But every once in a while, someone comes up with something and you go, wow, that's new. Look at that. Look at that horse coat. That's a little bit shinier than everybody else. And for some reason, because it's shinier, it makes me think as a female that stallion is probably healthier than everybody else because he eats better, right? If he eats better, it's probably because he's tougher, faster. Predators don't get him, whatever the case may be, right? Every once in a while, that does happen. But right? Formation of new alleles, that's real careful. You got to be real careful. You can change the number of genes or change the positioning of genes. This is something kind of interesting, especially where years ago we used to think that the introns were garbage. We used to call it actually junk DNA as if it meant nothing. Now, there still are introns, don't get me wrong, but we're finding out that we go into some of those, what we thought were introns, we're realizing that actually they're kind of like gene bosses. They regulate genes. They regulate the genes being activated or not activated. They may regulate how a gene interacts with an environment. We also have evidence when we look through genetic analysis that genes have been duplicated. I'll show you that some of the results of some of the research that's looked at this, and as I said, where you put those genes. We sort of discussed this the other day. One of the ways you can generate a genetic variation is reproduce a lot, right? I mean, every time you reproduce, you're doing it with a gamete, a sperm or an egg that has gone through, let's remember what's it gone through, that sperm or that egg, not mitosis, right? It went through meiosis. And I'm hoping that you remember from Bio 1 that meiosis had, yeah, there you go. Thank you. Yes. Yeah, it don't matter. It sounds right. I'm hoping you remember that meiosis, oh, I didn't see you come in. Hi, how are you doing, Leah? Has 2 big purposes, right? So the big purposes of meiosis, in case you don't remember, is we make these kinds of cells, diploid to haploid, and the other big purpose of meiosis, the reason why crossing over occurs, you want to create variation. Yeah, there's the connection back to Bio 1, right? So now, every time, sexually reproducing organisms do that, we produce a different type of sperm and an egg, and that means we produce different type of offspring. Even, like I said the other day, even among identicals. Now, remember what I said about asexually reproducing organisms. They're genetically alike, genetically clones, quote-unquote clones, ostensibly, they're clones. But of course, as I mentioned to you, even when you do mitosis, you do it a bunch, a bunch, not once, not twice, a bazillion times, whatever number that might be. Mitosis isn't exact every time. And most of the time, if it's a bad thing, something fixes it. A lot of the time, it's going to be in the introns. So nobody cares about it. So what? My junk mail is misspelled. But if it happens in an exon, and it just happens to be a good one during mitosis now, not meiosis, then that organism is going to be fit, just like if it had happened sexually. Now, how do you do that in asexual organisms, since the chances of variation are so small, right? Every time mitosis occurs, there may be a change. Every time meiosis occurs, there is. Crossing over. It's a done deal. It's an understood fact that meiosis is going to make variable gametes. Mitosis, you remember, is supposed to make identical cells, but it didn't really make identical every time. 1 in a bazillion, right? Whatever you want to say. If you reproduce a lot, you increase your chances of getting that 1 in a bazillion genetic variation that happens. And I'm going to show you today how that works in E. coli. You're going to be, oh my God, overwhelmed. You're going to be just floored by how many bacteria, E. coli bacteria, produced in a day. Did I ask you guys if any of you worked for Chipotle? Nobody works for Chipotle. Lots of people come in. Nobody works for Chipotle, do they? Oh, okay. Good. It's a good place. I made a comment last semester about Chipotle. What do we? Well, you'll see when I talk about the bacteria. But I mean, no fault really for Chipotle, in the sense that what they're trying to do is produce or has food that's natural and not preserved. But I mean, you know, that's the problem with that. The reason why we have the preservatives is to keep sometimes those bacteria away. But that's the balance you take, right? And then finally, and I've already sort of alluded to it, sexual reproduction produces variation, right? So that's the 4 ways we're going to look at the way variation works. All right, so let's take a look at the first one, formation of new alleles, right? Making new alleles, alternative forms of genes, right? Different letterings, different genes. So again, that thing that you got to be really careful, and that's why I put the dice down there to go, man, you're rolling the dice when you're doing this. Remember back from Bio 1, there were this thing called point mutations. And they're called point mutations because I always tell my Bio 1 students, because you can point to them. There it is. Where's the mutation? There. Hey, see it? It's supposed to be a T. Remember, this is what? Sickle cell anemia, right? It's supposed to be CTT. There it is right there. That point right there. That T got mutated somehow, whatever, to an A. Okay? So because that happened, you got a different codon. Because that happened, you got a different amino acid. Because that happened, you end up with hemoglobin that's not got the right shape. It's sickle cell anemia. That's why we call it a point mutation. Okay, now that's not a good mutation, right? Come on, sickle cell anemia, good or bad? Pros and cons of sickle cell anemia. Some of you might know something, though. Some of you might know something about sickle cell anemia, though. Does anybody know a little interesting fact about sickle cell anemia, or specifically? Yeah, isn't that interesting? We'll talk about that later. Yep. Sure does. Interesting, huh? Isn't that something? Yeah. Not necessarily all of them. We find out that it varies. That's why people with sickle cell anemia trait have to be careful. Because if they go to places where, for example, there's a thinner atmosphere, the lower oxygen levels might stimulate some of those red blood cells start sticking together. Oh, did it? In one of the labs, I did. Oh, in the when we were doing the Hardy-Weinberg, yeah. But I always find that kind of interesting how, you know, all this develops together. So we'll talk more because malaria is caused by a little protozoan. That's what causes it. And apparently having those sickle-shaped red blood cells, the protozoan cannot infect those as well. So that's why people with the trait have advantage. Not having the disease, but an advantage. Did you know that you hide genes? You hide genes. Well, all, you and your kitty cat, are the majority of mutations. What kind of mutations? Okay, partial edit. Yes. You can only adapt to what is here and now. You can't adapt to tomorrow. next year, or a million years from now. So you're only adapting to what's now. So when we look at big evolutionary changes that have occurred, we realize that, you know, that's going to happen through little tiny partial adaptations that move along the way. So yeah, is that what you mean, Alexis? Partial adaptations? I mean, by definition, an adaptation is not partial. It's only partial in retrospect when we look back and we say, oh, you know, when we look at the whales and how they came from a land ancestor, and we look at the land ancestors, and one of the things we notice, these guys with legs, okay, they're land, but they're whale ancestors. We noticed their skulls were getting longer. It's called telescoping, and their nostrils were moving from okay, the front, like a dog's nostrils, they were moving them to the top of their head, right? And that's where the blowhole was. We see that in organisms that were even land dwellers, but they were spending a lot of the time in the water, so it would be an advantage if your nostrils were up higher, right? Yeah, cool. Good question. We will actually get back to that. But yeah, it really is. It really, and it almost is like when people get this idea of looking at what's evolution's purpose, I'm like, none. Look at that. There's no purpose in any of that. It's just the mosquito trying to survive by sucking your blood, the parasite in the mosquito trying to survive by using your cells and the mosquito cells to reproduce, and of course you being bitten by the mosquito trying to survive. It's all this random thing going on, but it all kind of works out. It's what we say, that randomness, and then you look at how natural selection just kind of whittles that randomness down. It's fascinating to me. Come on, hidden genes. Who's got hidden genes? Right? Recessive genes don't show themselves, do they? Recessive genes? Typically they don't. There's some exceptions, I'm sure. But if you're a, let me type it in there. If you're one of these genotypes, that little A allele is not going to express itself, right? The only time it's going to express itself if you're one of those genotypes, right? You get that one, obviously you're not going to express it. So what that really means is that the little A allele there in the heterozygote, if it's the typical, you know, Mendelian way, that's hidden. What does he mean hidden? Well, Mother Nature has no way, as I just said, of knowing what the future is going to be. No way of knowing. That's why I wrote future use, question mark, ... blah, blah, blah. You know, but what can happen is, because the environment becomes some way, because the environment changes for whatever reason, it just so happens that if you have that recessive allele, okay, you're going to get some advantage from it, right? So that recessive allele, if it gives organisms an advantage in a population, it might actually be recessive genetically, but it might be more prominent in terms of the number of organisms in the population that have it. You see what I mean? Recessive doesn't mean minimum amount, right? Necessarily in terms of dominant recessive in genes. 47% of, it's probably different now, half of males of white Caucasian descent have O-type blood. You remember from Bio 1, that's homozygous recessive, O blood. O blood is homozygous recessive. Right? It's not, so that does, you know, and half, so that's definitely not the case, you know, recessive if you look at that. So what do I mean by future use? Well, as long as these recessive alleles, and if they're not expressing themselves, they shouldn't be a problem. As long as they're not making it worse for you, they kind of tag along. They kind of tag. Why? Because every time DNA makes copies, it makes copies of that thing. And if that little recessive, just kind of make, it's like if you had a spot on a page, you know, making copies on a copier. Oh, what is that? Every page has got it. Same thing, right? They're just kind of going along. And then something happens. Mother Nature rolls the dice, and here we got, oh my goodness, it got warmer outside. The temperature went up for whatever reason. Let's say volcanoes started erupting, okay? Caused the temperatures to go up, and now those organisms that are the little a, or at least the A and little a, you know, these guys, at least carrying one of these recessive alleles, you have an advantage, because that little a allele says, guess what? You got bigger ears, Bigger ears. And bigger ears means you cool off better. Like we show, I think we looked at the rabbit, didn't we? See? Now, okay, so you see what I mean by that? You see what I mean by the fact that Mother Nature can't predict what the future is going to be like, but those recessive alleles that are being carried along, they might sometime, like, okay, folks with sickle cell anemia trait, that genotype, let's say, right? So you don't have sickle cell anemia because that would be recessive completely. You're not completely not sickle cell anemia. That would be homozygous dominant, but you got the trait. Remember I said that you can actually suffer some of the symptoms in certain conditions. But those individuals were carrying along, those sickle cell anemia trait folks were carrying along a recessive trait that happened to end up being something beneficial. It was beneficial because along comes the mosquito, okay? And that's an environment thing. And you got these recessive individuals, and for some reason the mosquito's parasite has trouble, those guys. So, and I'm really kind of cutting ahead of the story, but if you're thinking, where's this ending? And I will give you a little foreshadowing. It explains why we see higher rates of sickle cell anemia in individuals of African or Mediterranean descent. Because, and I'll show you a map later on, you can superimpose the mosquitoes that cause malaria on the areas where you find high rates of sickle cell anemia, right? It's like a little bit of detective work. So that recessive gene in this case in that environment becomes beneficial. But there's no way that that humans could have said, I'm going to carry around this recessive allele because I know there's some mosquitoes down there that are going to be trying to suck my blood with a little tiny protozoan. Mutations are not common, right? But they're not rare either. We have what's called a genetic clock, kind of. It says that on average, for every generation, you know, cells dividing, organisms dividing, that out of every 100,000 genes, one of them mutates. Now, you don't have that many. You have 30,000, something like that, I think humans have. Somebody remember how many genes do we have? Look it up. Somebody look that up. How many genes do humans got? We did the Human Genome Project. So, I mean, I think it's like 30,000. So, oh, is that right? 20,000. Okay, 20,000. So if we look at that, humans, if you do the math, and I have to check my math now because I was assuming about 30,000. No, I think that's right. If you look at that, for the number of humans that are born on Earth, that humans create about 65,000 mutations a day. 65,000 mutations a day. Now, that one every 100,000 genes per generation is among all organisms on average. So on average, humans the world over, 8 billion of us, make about 65,000 mutations a day. Most of those occur in the introns again, so they're neutral and they're silent. So while you can get new alleles by things mutating, as I said, you know, oftentimes that's bad, the chances of you getting a mutation are pretty slim, and the chances of you getting a small mutation, or I'm sorry, a good mutation, a better mutation, an advantageous mutation are even smaller. It's like trying to hit the lotto, right? The lotto is, what, 6 different numbers, 1 through 47, so your chances of winning are 1 over 47 to the 6th, some insane number, something like that. See, and I also write down below, the problem with mutations, if they change the phenotype, usually that's bad. And if you affect the phenotype harmfully, then you reduce the organism's fitness. You make it less adapt. And don't forget Lamarck down at the bottom when it comes to mutations. Even if the stretching of the neck of the giraffes would have somehow mutated giraffe neck cells, those cells aren't the ones that are involved in reproduction. Chance, Aaliyah, there's a lot more of them than there are exons. So 95% of your DNA is an intron. 90% or something like that. It's probably like a mutation considered. No, I have an eye chiller cart, Sean, somewhere around here. In one of the classes, I think we go into that, or maybe it's a bio one. There's one. Look online. Look on Google. That you type in eye color chart. It'll show you all of the different genotypes in the eye colors. See, we used to think eye color was just one gene, but now we find out that it's different. Where's that chart? Hold on. There it is. Hold on. I'll show it to you in just a minute. As soon as I get the. Oh, that's not it. It shows all the colors, but it doesn't show the genes. Well, this is kind of what I meant here. Oops. So pull this up. So each of these, of course, are going to be different genotypes. So you take the genotypes of the parents, and again, it doesn't happen. You can find places where they do the genotypes. So if the parents have the genotypes that give you the 2 green eyes, and you got a 75% of a green-eyed kid. Now, purple, it doesn't have purple, but my guess is you could probably find something that's, because, I mean, these are color variations. You could probably find something that gives you purple eyes. Either that or it's contact lenses. You see some with yellow eyes. I'm like, I don't think humans have yellow eyes. Not that I'm aware of. It's mostly reptiles. And, okay. So again, the stretching of the neck cells of the giraffe, that wouldn't do nothing. It would changes, mutations that have to occur not in the somatic cells, the non-reproductive cells, but in the germinal cells, the cells that give rise to the sperm and the egg. Of course, this has to do with sexual reproducing organisms, right? So, again, are these ways that mutations are introduced in organisms that allow organisms to better adapt? Yes, absolutely. Don't for a minute think that this is saying it doesn't happen this way. It does happen this way. It's very dicey. Ah, wonderful, Steve. It's very dicey. It's very dicey if you get a good mutation out of this, though. But it does happen. We can't deny that. This is a source of beneficial, advantageous variation. Mutations because of variations. What was the other one? Oh, number, gene number or gene position. This is Cooper's nose down here in case you wonder whose dog that is. But if we change chromosomes, deletions, remember from Bio 1, insertions, duplications, all those things that you learned about, those are usually harmful again, right? Because Mother Nature is all about that balance of homeostasis. So yeah, if you delete a part of a chromosome, if you delete a part of chromosome that's an intron, no problem. But if you delete a part that's an exon, then that's a problem, okay? That gene's not there. And if you insert something into an exon, you scramble it. I always tell my students in Bio 1 that insertions are like you're reading through your bio book, right? Maybe photosynthesis, blah, 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 photosynthesis. And then all of a sudden you're talking about the Renaissance. And then you go back to photosynthesis. And on the exam, you're like, what does the Renaissance have to do with photosynthesis? Nothing. It got shoved in there. That would totally discombobulate the genetics there, right? Changes the code. Duplications can be good, but remember, Mother Nature, that short stick again, she's like, nah. You got to be careful when it comes to duplications because you start upsetting that balance thing. So while duplications can be good, gotta be careful with them. Translocations, yeah, I guess I could probably find online places where translocations have ended up in beneficial mutations, but remember that's where piece of chromosome is. What it is during synapses, during chromosomes crossing over meiosis, the homologous ones don't pair up. So instead of the 2 maternal and the 2 paternal, it's like a 2 and an 8. So you get chromosomes mixed up to different ones. Yeah, some of it too also is going to have to do with the light reflecting through the eye too. That's why blue-eyed folks who tend to have less of the coloring in their eyes are a little more variable. I know my wife says when I wear a blue shirt, my eyes look a little bluer, and when I wear green shirts, they look a little greener because the light that's reflecting off my shirt, if you remember from Bio 1, the color's reflecting off is the color it is. Hits your eyes and then bounces out too. So purple might be one of those things where it's a color, then a combination of where you are in the light too. Now, while translocations, actually, that's right. I didn't think about that. Translocations and changes to chromosomes are usually harmful. But, if a translocation moves genes closer together, especially genes that kind of do the same thing. Remember you learned in Bio 1 that it's not one gene, one enzyme, boom, we're done. Although it can be. Well, I don't think it's one enzyme, but very often one gene controls making one part of the enzyme or one aspect of the enzyme, then another gene controls another part. Usually it's a collaborative event of genes. There are several genes. Most genetic traits are polygenic, right? Polygenic. Many genes that control one trait. Human height, hair color, all that sort of stuff you learned in Bio 1. Yeah, I think it can change, Aaliyah, because I think I was reading not too long ago that the melanin or the coloration in the eye can change throughout your life as far as how much is deposited or not. I'm not sure it can flip like brown to blue, but I think, you know, it can change in within that parameter. I think they've got it down to like, I think not 3 genes that actually control eye color. So all that big B, little b stuff that you learned in Bio 1 wasn't exactly right. It was kind of like the atom, you know, the more predictor. One with the genotypes. Okay, so if you put genes that are far apart that do a similar job or are linked together in terms of function, you put them closer together. It's like moving a factory closer together. They're more efficient. That can be helpful, okay? Can't make large duplications. That would really upset the balance, but you might inch in. And the nice thing about small duplications, as long as they're beneficial, you can keep adding a little bit, right? Our ancestors, our mammalian ancestors, had one olfactory smell gene, just one, right? Humans, we got about 350. Not a bad sense of smell. Mice have about 1,000, 3 times as much. And Cooper the dog, he has about 3 times as much as well. 838 olfactory genes. So as I always tell folks, dogs smell the world the way we see it. They smell it with that kind of depth. So when you come home, your dog knows where you've been, what you've eaten, who you've talked to. Their sense of smell, that's like when I take Cooper to the beach. He just loves it because, you know, he's living inland here in Oviedo. So when I take him to the beach, he's like, oh my goodness, all these new smells. He's all in the seaweed and all that kind of stuff. It'd be like us in an art museum, right? Oh my goodness, look at all the beautiful paintings. And elephants have a lot more. Elephants have an incredible sense of smell. Very good sense of smell. Twice as many as dogs. When I worked for SeaWorld, we used to go over to Busch Gardens, and a friend of mine was running the elephant exhibit there. And one of the things they used to give the elephants to do for fun is they would hold out big broomsticks or wooden sticks with paper towel rolls on them. The elephants would take their trunks and grab the paper towel. They love grabbing the paper towels, whipping it up in the air, throwing it all over themselves and walking around. They just thought that was something to walk around with all this paper towel rolls hanging all over for some reason. I used to watch them just crack up and think, oh my goodness, what is so fun about that? But for an elephant, that's just woohoo! That's a party. Nah, wasn't that, because there was plenty of shade around. Albinism is a recessive disorder, so folks like this, no big deal. Carriers, somebody who's albinistic. Now, an albinistic individual, the reason why albinism has such wide-ranging effects, remember, is because it's the opposite of polygenic. It's pleiotropic, which means one gene controlling several traits. So if you're an albinistic individual, you're going to have reduced or no melanin in your skin, and your eyes, in your hair. It's going to affect all of these things. So, and again, that would be recessive. Why would albinism be recessive? This is a good little chance to do a refresher about what we know about evolution. Why would albinism be a recessive trait, would you think, in this particular case? Well, yeah, that's right. Can you think what the problem would be, though, that would cause these individuals not to be able to be as reproductively fit, let's say, as other individuals? That is, not live as long and have as many children. Yeah, the mutation is uncommon. Yeah, that's something that's interesting that you bring that. I wasn't thinking that in my head, but that's absolutely true. That in certain animals, And albinism can occur in plants, actually. But in certain animals, albinism would definitely be difficult for them to blend in if they were trying to hide from prey, or if they were predators, stalking predators. You know, the reason why your skin is the color it is, right, and you could go out and you get a tan is because of that stuff called melanin. And folks who have albinism don't produce melanin. Well, melanin doesn't just give you that nice, lovely bronze tan. What it does is it absorbs UV rays. So UV rays are bad for you. Bad, bad, bad. And our ozone layer does a lot of that, way up high. But with still a bunch get through. That's what burns you at the beach. UV causes cancers. DNA mutations, messes things up. So melanin acts like a buffer. Folks with albinism, kind of, you know, they're not going to want to live in Florida. That's for sure, right? A lot of sun. I do remember a student when I adjuncted at Valencia, though, that was an albinistic student. She just always wore shorts, and or not shorts. She always wore shorts. She always wore shirt sleeves. You know, she always had sleeves on her shirts. Yeah, she always wore short sleeves. That's it. Rapid reproduction, right? Now, mutation rates in animals and plants is low, and I know that some insects are crazy and have hundreds or thousands and all that sort of stuff. You know, these mutation rates in prokaryotes are often lower, which kind of seems counterintuitive, right? If mutation rates in animals and plants are low, and sometimes are often lowered in some prokaryotes, why is it that prokaryotes can take advantage of rapid reproduction to get variation and we cannot? Well, remember, prokaryotes have no introns. They have no junk DNA. There's no such thing as a silent mutation, right? There's no such thing. So when you couple that with a relatively high reproductive rate, right, maybe sometimes not as big as animals and plants, because every single change affects the critter, you get a lot of different critters. So I did a little math there for you. There it is, E. coli, right? The intestinal bacteria that causes problems if it's in the wrong strain. I tell my Bio 1 students, you have E. coli in your gut right now. It's just the strain. If you have to get the wrong strain, then it makes you sick. So E. coli on average produce 2 times 10 to the 10, and I'll let you figure out what number that could possibly be. Offspring a day. So they divide. The spontaneous mutation rate among these organisms is about 1 in 10 million. All right, so 10 to the minus 7. So we do a little quick math there. I'm not real concerned about you following the math, that's really the end result, but I want to show you that with that such a high reproductive rate, even with a low mutation rate, 2 times 10 to the 10 cells, that's not even a number, right? That's just some big number. That you don't even have a name for that thing, can't. Even though the mutation rate is only 1 in 10 million, that still produces, since every single one is a physically expressed mutation, there's no junk DNA. You get 2,000 bacteria a day with mutations. 2,000 non-silent mutations a day in bacteria, you see? So if you're reproducing relatively high, even if your rate is lower than lots of animals and plants, okay, you can still take advantage of that because you're producing so many offspring, right? Isn't that neat? It's like throwing everything up against the wall to see what sticks. Let's just produce lots of offspring with this high reproductive rates. You know, even though the mutation rate that we have amongst our DNA might actually be lower in animals and plants, we know that every time we get a hit, every time we change a letter, we get a change. Now you know that the genetic code is redundant, right? The wobbly base thing. Okay, that's fine. But still, you increase your chances of getting a mutation. And listen, remember from Bio 1, all you got to do to get a mutation is change the amino acid. I mean, yes, we're changing the DNA. That's the definition of mutation. But the end result is, what does it do to the amino acid chain? If it doesn't change it, like the wobbly base, who cares? It's a silent mutation. Again, it's another type of silent mutation, even in an exon. If it does change the amino acid, maybe who cares? Because if it changes one amino acid for one non-polar one for another one, may not affect the folding of the protein. But if it switches a polar for a non-polar, you get sickle cell anemia. See what I mean? All right, what's the last one? Oh, yes, sexual reproduction, okay? Sexual reproduction doesn't apply to everybody, If you're like many prokaryotes, or all prokaryotes, you're going to engage in asexual. But for those of us who engage in sexual reproduction, let me remind you once again, that's sperm and egg. That's the definition, right? For those of us who do that, when we cross over, see that? Isn't that a cool picture? It braids. This is actually two chromosomes. Now, crossing over occurs at random, obviously, every time the chromosomes get into synapses. About, I believe, 2 to 3, less than a half dozen different crossing over points each time, all variable. Then, of course, if you look down below, and excuse me. Hold on one second. Let me Google. Sorry about that. So if you look down below, we don't need to memorize or put this chart to our brain, right? I'm reminding you that in meiosis there are two, or actually many ways to get variability, right? The crossing over, because where they cross over, then they separate in anaphase 1, so that mixes all that genetic information up. And then when they go through meiosis 2, remember Mendel found out Mendel's second laws. They assort independent of one another, the alleles do. So what happens, what ends up in one gamete has no, genetically, has no effect on what happens, ends up in another gamete genetically. So it's all random. The whole process of meiosis is blah, blah, rolling the dice, randomly where they cross over, randomly what cell they end up in, randomly in the end there, right? Randomly which cell gets fertilized or does the fertilizing, right? That's all random. Organisms don't have any control over that, really, right? They have control over mating, perhaps, with a particular mate of a stature, if you will, going back to our horse example. But then when it comes right down to it, the sperm and egg getting together, you know, the egg you ovulate, you can't say, okay, this month I'm gonna ovulate a kid with blue eyes. Okay, there we go. Even if you had that genetic information in you, and as a male, you can't say, okay, guys, it's gonna be this sperm that does it. I mean, we can't do that if you engage in fertilization in the traditional way. Obviously, there are ways in the laboratory to get around that sort of stuff. But all of that is creating a lot of genetic variability, right? Crossing over, then sorting out the alleles into different types of gametes, not different types, but gametes differently genetically, then fertilization being, and what do we end up with? We end up with just a whole bunch of everything, right? Is that good or bad? To end up with a whole bunch of everything. Why? Why do you say that? Why do you say that? Yeah, and you're good. Yeah, can't avoid that. Remember, Darwin said that we can produce more offspring that can possibly hope to survive. So what you guys are stating there, yes, that's Malthus again, inevitability. Of course, things are going to clash. So if things are going to clash, think back again to my original question about, is all this variability, is it good or bad? Things are going to clash. Would it be good for the species if it wants to survive into the future, which they typically do, if some of those variations gave them an advantage? So while everybody is clashing, if you will, maybe somebody clashes better than the other one, reaches up higher for the leaves at the top of the tree, runs faster, whatever you want to, whatever you want to think about. Yep. I mean, yes, it is a struggle for existence. Darwin called it nature, red in tooth and claw. It's a cool little saying. Nature, red in tooth and claw. I think it was Darwin. Basically saying that it's a dog-eat-dog world, and y'all are wearing milk-bone underwear out there. And yes, there is a struggle for existence. We all know that. But remember what Darwin's key part of the theory was the fact that that struggle for existence means that those who have an advantage, doesn't have to be a big one either, those who have an advantage are the ones that are going to reproduce more, leaving behind more offspring. That's how organisms perpetuate into the future. Now, organisms would never have to evolve if it weren't for the world changing. If the world stayed the same, climate, predator, whatever environmental variable you want to pick, if that never changed, there would be no, if it ain't broke, don't fix it. Organisms are not going to evolve. They don't need to. It's the world changing. Which, remember all the geologists saying nothing you do about that. It changes, right? It erodes and mountains form and climates change. And we're going to talk about it in later chapters. But there's nothing you can do about that. Nothing. The only thing you can do is just keep throwing variation out there and hoping that you have the right variation to keep going into the future. Sometimes you don't, and you end up like the dinosaurs, dead, gone. Oh, gosh. Hardy-Weinberg. Oh, yes. Have you all been playing with the Hardy-Weinberg problems? Please tell me. I would say everybody shake your head yes. Yeah, good. Please work on those. If you look online, I posted something, didn't I? Yeah, I did. I found something online. I posted a link to it with some practice problems. They're not hard. Practice. Follow that spreadsheet. Follow that flow chart that I showed you in lab, and you'll get them right every time. Okay, just get, and even if you think you know how to do them some more, because eventually what happens is going to be you get in, or not eventually, but what gets in, inevitably what happens, students get into the exam and they freak, right? And they go, oh God, what is it? P squared, Q squared. Good, right. And go online and find your own too. And if you find some good ones, post them in messaging. You can message. I think you can message the whole class. If you can't, send it to me and I'll send it out there. Okay, so you've been practicing that. So let's get back to the idea of what it's saying. Remember what you're calculating there are allelic frequencies. Allelic. So what you're calculating is in a population, how many are this way? How many are homozygous dominant? How many are heterozygous? How many are homozygous recessive? You're counting that. Now, Sean asked the question about, aren't we also looking at the frequencies of each allele? Yes, you are. We're not only looking at the frequencies of the genotypes, but we're also, of course, looking at the frequency of just the A or just the little a in a population, right? Works the same basic way. Now, as you remember, the thing with the Hardy-Weinberg, why it's necessary, if you will, is because we don't know that. Oh, shoot. Scratch the second one. There we go. We don't know AA from AA, right? We don't know that. You can't look at something and say, can't look at a brown-eyed person and tell they're heterozygote or homozygote. So we use the Hardy-Weinberg equation to calculate all those, okay? And that's why we always say, what's the first step in the Hardy-Weinberg in the flow chart that I gave you? When you look at a Hardy-Weinberg problem, what's the first thing you do? There you go. You read in that problem, says, where is he telling me how many have the recessive phenotype? Now, I may not say 30 out of 100 have the recessive phenotype. I might say 70 out of 100 have the dominant phenotype. You know, and then I get emails, oh, Mr. Clark, I'm like, subtract, okay? Think, right? So, exactly. You got to have Q squared, right? So once you got that, that's the recessive phenotype. Then you can calculate all the other values. Now, you've done that, but I want to go back again to what that is telling. So we get these numbers. The Hardy-Weinberg equation, or really the Hardy-Weinberg equilibrium, says this thing. A population that's in equilibrium, that is not evolving. Okay, so we're demonstrating a point here kind of in reverse. So if you're getting a little confused, think about that. We're proving something evolves through Darwin's methods of natural selection by disproving that the change occurred through something else. Remember, evolution is a population changing. The mechanism of natural selection is one mechanism. You're going to find today there's other ways to change a population, to evolve it. So when someone says evolution, it's what mechanism? Because is it Darwinian evolution? That's why you hear that term, Darwinian evolution. Why do you got to say Darwinian evolution? Because there's other ways it can happen. All right. Now, Hardy-Weinberg says none of that ever happens, no evolution, as long as these 5 conditions are met. In other words, you're not going to have any mutations that affect anything, right? You're not going to have those mutations in the exons that are going to affect things. And you think, well, doesn't that happen? Right. And that's mutation would, if it's in the future, would be something that we would call beneficial, right? Remember, they're not usually. The random mating part means that the organisms are gonna make sure they keep that variation going. We're gonna not have inbreeding. Inbreeding is gonna affect the population, and it's gonna change the genetic information. Inbreeding, we call it bottlenecking. It whittles down the genetic information. It's why, next chapter, most all cheetahs in Africa are genetically identical, even though they're not related to one another, because the cheetah population was whittled down to so few cheetahs. Those that were left were very closely related, and the population that recovered afterwards is all very close. It's like they all came from the same basic stock. So random mating is not, you got to have random mating. Now, again, like I did in the lab, skip number 3 for a second. You got to have a really big population, okay, so that you don't have genetic drift, right? What's genetic drift? So let me skip ahead a few slides. Don't freak out. This is the Hardy-Weinberg thing again, everything we talked about, right? See, the Hardy-Weinberg equilibrium, once again, there is actually good information on here. Remember that phenotypic variation reflects genotypic. What the heck is he saying? Yeah, it is, Sean. It's awful for the cheetahs, too. It means if anything affects one cheetah, you know, disease-wise, probably going to lose them all. So phenotypic variation is a reflection of what genotype you have. Big A, little a, right? You all know all this. So if we change genotypic frequencies like we're talking about in the Hardy-Weinberg, the population is evolving, like I said. But I indicate in red, once again, that's not necessarily through natural selection, all right? It's not necessarily. Now, I'll come back to what this microevolution term means in just a second, but let me jump up and show you some other ways that it can change. There's the formula, okay? There we go. All right. Now, you notice that the first one is natural selection up there in the upper right. So, yes, absolutely. Like I said, evolution can work through Darwin's theory of natural selection. That's the way adaptation occurs. But let's go to the, let's jump ahead to genetic drift. We'll come back to natural selection. All right. Here's what I mean by genetic drift. Genetic drift, what decides the change in the population is random chance. Are you thinking, wait a minute, that's what decides it in natural selection? No. Variation comes about through random chance, but organisms, as they adapt, those that are most fit, there's nothing random about that. You're either more fit or you're not. You either can reach up there and get that browse at the higher place or you can't. You can either outrun that predator or you can't, right? There's nothing random about that. But in genetic drift, random fluctuations, in statistics, we call random fluctuations, or sometimes you might hear statistical noise. We call that drifting because it's just drifting back and forth. But random fluctuations in the allelic frequencies, if they happen in a small population, a finite population, that can have an effect, right? But the problem with this, with genetic drift, the reason why it's not evolution in the Darwinian sense is it doesn't have anything to do with adaptation. Let me show you what I mean. We got, 10 flowers there, right? We go through, and like Sean was pointing out in class, we can actually get the frequency of the red alleles. And in this case, it's 70% of the alleles are red, 70% are white. We're dealing with those incomplete dominant plants you learned about in Bio 1, remember? Homozygous dominant, you're red. Homozygous recessive, you're white. If you're heterozygote, you're pink. Right? Remember that was one of the plants that when biologists first looking at genetics kind of screwed them all up. They went, oh, inheritance must be blending. All right, so anyway, here's what we're talking about all of this. It says here 5 plants leave offspring. Now, we don't mean that those 5 plants are hardier than everybody else, but let's say the plants that are not in the yellow, all right? So ignore the yellow plants. So it's the 1, 2, 3, 4 red ones and the pink one there. Let's say they are at some part of the edge of this beautiful mountain meadow of flowers, right? Here we are in the Alps. And we got these red ones and pink ones and white ones. And it just so happens the ones I pointed out, the ones in the white, they kind of, more of the red and the pinks are kind of over in this corner for whatever reason. Boulder comes down the hill. Boo, ba doo, ba doo. There's an avalanche. Rolls over those flowers and kills them. Now, first, before we go on, the boulder rolling downhill and killing the flowers. Is there any way you could blame the flowers, right? Did they fail to adapt? Is there any way that we could reasonably assume a flower to adapt a mechanism deal with rolling boulders, right? No, gosh no, right? So it's random chance that killed these things. Yeah. I think about this when I'm in the woods. I like to go hiking a lot in the woods, and I think about this as I step over a fallen log. Sometimes I'll step over and I step on a thing of moss, and certainly I kill some of that moss. I think, oh, drift. Those poor moss. The moss I stepped on, for all I know, could have been the best moss. They could have been the fittest moss. They could have been the champion moss. But just because of some clumsy-footed human stepping on them, they're gone, right? So it's all about, in genetic drift, it's all about chance. If you see a question on the exam that asks you about genetic drift, think chance. The controlling factor is chance. It's not that of natural selection, right? Now, as I said, it's all about the finite population. The smaller the population, 10 flowers versus, I don't know, 10 million, and 5 get killed again. Let's say same thing, 5, right? It's not going to have quite the effect on that big population that it's going to have on a little population, right? See, the next time around something happens, maybe, oh, what happens this time? Let's say some grazing critter comes through and just randomly picks and only leaves the red ones. In 3 generations, we've gotten rid of all those other alleles. That population has evolved. Yes, true. It has evolved. So if I asked you on an exam, did that population, you say yes. Mechanism, not natural selection, though. Random chance. Down below, I give this little neat little graphic or statement. The genetic drift on population, the effect is inversely proportional. That's what that little squiggly thing is. In case you didn't remember that. So what that means is the bigger the population, the less the effect of genetic drift. Makes sense, right? See, natural selection's not that. Let's go back to, there we go. In natural selection, you got this owl coming down feeding upon these mice, and they're all the same species, but, you know, mice are born all of the same species of different colors, just like the horses and everybody else is. Yeah, I might. Or I might ask you to explain the different ways that things can change, or I might directly come out and say, tell me why drift is not natural selection. See, the thing that really you got to keep in mind is the end result a fitter organism, and in drift it's not. It just so happens the ones that were in the wrong place at the wrong time with the rolling boulder. In natural selection, like in this case, it's the fittest organism, right? So this owl that's preying upon these mice that are of a bunch of different colors, it just so happens the mice that live on the basalt rock there on the left, this is actually from some real data, the mice that live on the basalt rock, the ones that are darker, that's the darker rock on the left-hand side, they're the more fit. They're favored by natural selection simply because the owl can't see them. So the light-colored mouse in this particular case is not as fit because he's going to get polished off quicker. But if you take the same species of mouse and you take a look at them out into the desert area, everything flips. Now the light-colored mouse is favored, okay? That's all about adaptation. If you're saying, okay, why is that adaptation? Adaptation means that you efficiently use your environment. It's not a bad definition. Adaptation is you efficiently use your environment. Okay, so in the case of the dark-colored mouse, they're efficiently using the camouflage aspect of their environment. Giraffes were efficiently using the fact that they could reach up higher, the browse that was at the higher place. You see what I mean? So the end result of adaptation is organisms getting better adapted, getting better suited to surviving their environment. And then Mother Nature comes along and changes it all. It's almost just funny, right? As soon as all these mice figure it all out, everything changes. Let's say the weather changes in a few thousand years, also it starts snowing there. None of them mice got an advantage then. So that would cause change of a natural selection, but it doesn't for adaptation. This will cause change, but it doesn't because of pure chance. Pure chance. The last one, go back to the first, is probably the easier one, the easiest, gene flow. And that's organisms that move between populations, right? So that would cause that. Let me stick with drift, though, for a second, because there's some really neat things that come out of genetic drift. Again, chance. Always think chance when I say drift. Things are drifting. If something's floating down the water, it's kind of randomly. All right. There's something called the founder effect. And this is real important because in the next chapter, we're going to talk about how we go from, see, all we've talked about so far, there's that term, is microevolution. Microevolution is the population is changing. It's getting darker colored, the more darker colored mice, more lighter colored mice, more bluer beads, right? Red and blue, yeah. More bluer beads that we did in lab. It's just, but we're not making a new species. We're staying in the same species. We're just kind of playing with the variation within the species. But in the next chapter, we're going to go into macroevolution. And macroevolution is how do we get a different species, right? So we're not there yet. But the founder effect is a big part of that. And here's what happens in the founder effect. The reason why it's called the founder effect, it's always easy to remember, is because the new population of critters, okay, comes from just a few, so just a few founders. Now, if you remember Darwin's finches, says here I got bad connection, so if I am breaking up, please someone let me know, so I don't know. All right, so here's the mainland, Darwin's finches, South America. Here's the mainland. This is the mainland of South America. Now, and this would be the Galapagos, the islands, if you remember my example. Remember that? The 15 or 17 species? I don't want to write over that, what I wrote there. The Galapagos Islands are right there. So out on the mainland, or on the mainland, we have this. This is now one species of finch. Just one species of finch right here on the mainland. But there's variations. Is the video okay too? Because sometimes if I turn the video off, it'll help. Okay. So you get what I'm saying? All of these are finches, but there's finches, and the different colored dots are going to represent variation in finches. Whatever, beaks, feathers, whatever variation is there, you got 3 of them, okay? So here's what happens. By chance, actually by luck, just chance, right? My wife, a while back, tells me, she goes, I had a dream last night that I dreamt 3 of the lotto numbers. I'm like, what good does that do us? Like, dream the other 3, then let me know. But 3, well, you don't even win $5 with that, do you? Right? So you can go there and bet all you want on birthdays and all this stuff, but you could bet the numbers 1, 2, 3, 4, 5, 6 and have the same chance of winning. Take a statistics class, you'll figure that out, right? So these birds get lucky. They get, maybe there's a tailwind, a storm, all right? Or maybe it's the seagull that reminds me when I was in the Navy when we left San Diego. It left San Diego with us, and for days we're heading out to sea, and the seagull flew with us. I'm like, dude, don't you got to turn around sometime? He didn't. He just kept flying with us. He would eat the garbage we threw over the back, and then he would hang out on the ship when he got tired. He'd land on the ship, and then a couple of days before he got to Hawaii, he flew off. I guess he was hitching a ride, but it was only one of them. Probably not very common for them to do that. So these birds, for whatever reason, luckily, make their way over to the island. Why do I say luckily? Because most of the time they don't make it. See if I can draw a skull and, oh God, it's supposed to be a skull and crossbones. Most of the time they don't make it. Most of the time they fall into the water. Daggone it. Maybe. I don't know. Maybe birds develop, maybe that's an adaptation. They adapted to their environment, which were these big things that they could help. Because there's no way a seagull could make it all the way. Well, I don't know. They can float in the water for a while. I wonder if they could make it all the way. That is supposed to be a skull and crossbones, folks, because what I'm trying to illustrate is most of the time. Yeah, that's right. That's right. Well, yeah, that's what the problem we have here in Florida, right? That's where all those Mediterranean reef geckos come from. They come on ornamental plants that come over. That's where the brown anoles, the Cuban anoles came over on plants and things from the Caribbean. Absolutely. Rats. I remember being out in the middle of the ocean and seeing these huge piles of garbage, unfortunately, and there'd be these pallets floating. And I'd look through the binoculars. There was something moving on the pallets. It was all these rats. And we always just said the nearest land was straight down. you know, because we're way out in the middle of nowhere. Thinking how many of those rats survive. But so that's another good, we could use rats here too, right? So rats, rafting. Rats are really good rafters. They can last for a long time, but I don't think they can last forever. These guys were in the middle of the Pacific. I'm like, good luck, rats. But they could, by chance, make their way over to the island. So let me get all my scribble out of the way. But depending on who was the lucky ones, you could potentially certainly change the genetic variation. Now, I haven't counted up the number of reds and the blues and the whites and all that sort of stuff. But let's just assume that the proportions we see on the island, which we could do with our Hardy-Weinberg. So imagine the reds are the homozygous dominants, and then the blues are the heterozygotes, and the whites are the homozygous recessives. The proportion could easily be changed by those few. It's like how much is genetic drift. See, that's why they're the same. Those few founders that make it over to the island, right? They're not necessarily representative of the genetic variation. Ernst Mayr actually in the quote says it wonderfully, right? And the big thing is in the red. What if the founder was one fertilized female, right? One female bird with an egg, and they fly over to that island. How much genetic variation, what proportion of genetic variation from the original population does that one fertilized female have? Not very much, right? And the reason why we say this is, yeah, absolutely. So, you know, this is not adaptation. Now, be very careful, right? Because what happened with Darwin's finches is once they got to the island, then natural selection took over. But you've got to remember, many times it's important to understand, how did the initial change in the proportions happen? Okay, in genetic drift, the change in the allelic proportions happened because of chance. In natural selection, it happened because organisms adapted. So what happens with genetic drift, usually founders die. They don't make it. Or they land on the island, they can't adapt, they die. But if they survive and reproduce and perpetuate and all that sort of stuff, then they switch, then they done natural selection. Then their population evolves through natural selection. See the point I'm trying to drive to you there? What caused the initial change in the proportions? What caused the initial differences in the Hardy-Weinberg numbers? Was it something that had to do with fit organisms reproducing more, or differential reproductivity, Darwin's little thing about there, or was it due to just because you got caught in a windstorm or you happen to be on a pallet floating across the water? Okay? Yes, I understand that the rats floating on the pallet across the ocean might be better adapted at dealing with those kinds of harsh conditions, but let's just assume that it's not. This is just a chunk of rats. Chunk, herd, pod, bunch of them. Now, we say that this whole thing of the founder effect is more common on islands, especially islands in archipelago. You guys are really smart. You knew what an archipelago was. But the Galapagos are one of those little collections of islands. So that founder effect could be repeated many, many times over and over. A lot of differences there, wouldn't you? But it's not evolution. The initial, right? If those organisms survive and reproduce, then it's going to be evolution through natural selection. But then the evolution initially was due to chance. I mentioned the cheetahs before, kind of the same thing. And we can illustrate that. We call it the bottleneck effect because you think about a bunch of variation, the reds, what's this time? Yellow, right? Yellow and the blue. And then something happens, turn the bottle over, and there's some drastic reduction in the population. What happens? Some predator moves into the population. Humans show up. Something happens relatively quick. So in this particular case, you're going to whittle that population down very quick. Going to cull it. I mean, we're killing. And by chance, since there were so few red ones left, and the surviving ones that'll live are only the blue and the yellow, right? That's why we call it a bottleneck. Now, what caused the drastic reduction in population and caused which organisms to survive or not was not adaptation again, right? It was pure luck. It was pure luck. It was pure genetic drift. All right. Problem with bottlenecks, like I said, is this: maladapted. So organisms don't adapt; they become maladaptive. Right? So because of inbreeding, because of the fact that when you whittle down a population, so you take a genetically diverse population represented by these colors, and some bottlenecking effect occurs, and it could occur for a while, so you continue to whittle. If the population survives, and if you're thinking, gosh, they probably go extinct, they do probably go extinct. But if they survive, then what reproduce coming back rather is a very genetically simplified population. Now, again, what causes that bottleneck are things that not necessarily that we would associate with adaptation. So among the cheetahs, for example, climatic changes that occur during the Pleistocene, all right? So the climatic changes that occur during the Pleistocene, you might say, well, that's adaptation, right? The environment is changing. But what we're talking about are changes that whittle down the population randomly. So there aren't things that were having to do with whether cheetahs were adapted to the population or not. It just happened to be where they were at the time. It's kind of like those flowers. When we moved in the 19th century, anthropogenic means human caused. If you're up on the global climate change, and I hope that you are, it's important because you all are going to have to deal with this planet long after I'm dead. But global climate change, you understand what we mean by anthropogenic factors are things that we dump in the environment. Carbon dioxide, emissions, just all that stuff that causes warming of the planet, changes in the remember, it's not just global warming. Global climate change means changing the climate. Human beings do that. So when we moved into Africa in the 19th century, we said, wow, cheetah's pretty, we're going to shoot them and make coats out of them. So at least they died for a good reason, right? The biggest reason why critters die, though, yeah, Pleistocene. But again, it had more to do with where these guys were than basically being adapted or not. If you just happened to be in the wrong place at the wrong time, it killed off a bunch of the cheetahs. Today, in zoos, cheetah exhibits in zoos, it's very commonly known amongst the zoo veterinary group of people, industry, whatever you want to call it, that performing surgeries on cheetahs is not difficult if you're doing things like skin grafts or anything like that. You can take donor skin from one cheetah and graft it onto another without fear of tissue rejection because they're so identical. So they're practically identical. Now, that's a bad thing, right? That's a bad thing. For example, in the early 1980s, feline peritonitis almost wiped out the cheetah population because of a poor immune system, because of inbreeding depression. Could be. Yeah. See, again, yeah, Sean, a lot of times when we talk about a bottleneck, we're talking about how fast did this happen. Climatic changes during the Pleistocene that killed off the cheetahs happened relatively quick. It's kind of the cheetahs that got killed off just sort of got caught in the wrong place. So, yeah, when you think about moving into America, the colonists and especially the intentional spread of disease, then yes, whittling down of the Native American population could be considered a bottleneck. Absolutely. I'm not sure if you'd have to show that genetically, but that definitely could fit the criteria for that. Northern elephant seals, look, in 1890 killed them to 20, not 20,000, 20 animals, and now we got 150,000. Now you do the math. From 1890 to 2017, the number of years from 20 individuals to 150,000. You can look at their generation time, blah, 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 blah. You know what's going on here. You know that those northern elephant seals are genetically identical. There's very little genetic diversity. So again, whatever kills one is going to kill them all. There's no variation. See, this is the opposite. Right? These kinds of changes that occur. Now, this is a population bottleneck again. Why? Because humans moved in and we killed them really quick. It's to say, well, organisms die because humans moved in. Well, they didn't adapt. Well, come on now. You can't. It's like the boulder rolling downhill on the flowers. Give them a break. The flowers can't develop armor coatings to protect themselves against boulders. It's not going to work that way. That's why genetic drift in the bottleneck is chance, even though it's like, well, wait a minute, isn't this an environment changing? Yes, but come on now. This is environment changing in such a way that you basically are catching organisms kind of out in the open. Like, ooh, like the cheetahs. Oh, gosh, got real cold real quick. Okay, well, we got, gene flow. Where are we? Yeah, gene flow, last one. So gene flow is another way that it can change, and it's exactly what you think. Organisms moving in, organisms leaving are going to take genetic information in and out with them, and that could change it. And that's not evolution. Remember, I gave you the example of the fish in the lake the other day. Well, let's imagine there was another lake nearby, and those fish were separate from one another. There was a land in between, but a once in a 500-year storm came along and flooded the lakes, so they mixed. That would be gene flow, wouldn't it? And what are the effects? Well, it depends on what goes in and what goes out, right? Depends on what kind of what genetic information. My wife and I went to New York a number of years ago, and we went to Ellis Island, and I got this picture offline, but I remember there was a picture there in the place where they process, and I just thought, wow, what those people just standing in line, and all of those folks coming together, what that must have been like. And then you go outside and you try to find your name, you know. It's like, I don't know my ancestors. Oh, heck yeah. Sure, organisms are moving. There's also probably a lot of local extinctions, because if you think about some of those critters that live near the coast, they're used to not salt water. They might be used to brackish water, but they're not used to that kind of salt water. So, yeah. All right, folks, so this is a good point to end, but I want to look at this last slide. So here we go again, right? What causes evolutionary change? Drift, flow, drift, chance, flow, critters moving. That's not adaptive. The only evolutionary change, Hardy-Weinberg, the only one that is natural selection, that's the only one that's purely adaptive. So stick with me for just a second. I'm sorry. I meant to start scrolling back before. But if we go back to the Hardy-Weinberg equation again, the conditions of the Hardy-Weinberg, here's why we use the Hardy-Weinberg to demonstrate evolution by natural selection. Because we say it's not drift, okay? It's not gene flow. The organisms are mating randomly. There's not a source of, that's not a big source of mutation. So if the numbers changed, bingo, that's the only thing left. So you see what I mean? We use the Hardy-Weinberg to demonstrate natural selection is working by default. We say, okay, population has changed here 10 years later. What did it? Mutations? No. Random mating? No. Population is really big? No. It wasn't drift. There's no gene flow, no evidence of it. It's got to be selection. Sorry, I went over a minute. So y'all have a good weekend. I will see you. You guys are second day. So Wednesday next week, right? Monday is Labor Day. Please work on your homework assignments. If you have questions over the weekend, the holiday, all that, by all means, please email me. You'll very often get a response from me. So if there aren't any other questions, y'all have a good afternoon. And like I said, have a good vacation. Or if you have time off, if you do it all. Take care, Sean. See you later. I'll post this recording later as soon as it's ready. Thank you, Patrick.